Okay. Good afternoon, everybody. You're very welcome to this, I think it's the final lecture of today's sessions. My name is Gavin Davey. I'm from the School of Biochemistry and Immunology. And I was an undergraduate student here. I studied biochemistry back in the 80s. I did a PhD here and then went away for a few years and came back about 15 years ago and have been here ever since. I do a lot of lecturing in biochemistry and neurochemistry. Particularly my research interests lie in brain diseases mainly, but I also do a lot of lecturing in other types of for other types of student courses. We do a lot of research in the School of Biochemistry and Immunology. Indeed, it's probably the biggest school for biomedical research, one of the biggest schools for biomedical research in Trinity and indeed in the country. What I wanted to do today in this short presentation is to give you some background information on the degrees that we run and that we specialize in, why biochemistry is important, why immunology is important. And then, rather than give you too much of that type of information, I thought I'd just give you some examples of the type of research that we do in the School of Biochemistry and Immunology. Because really these examples will show you just how important biochemistry is in biomedical research. Now, we have tracked our students over the years. We're an old degree. Originally it was just biochemistry. Then as we got bigger and as college changed, we split our degrees into biochemistry, immunology, molecular medicine, and human health and disease as well. So I'll tell you about all of them. But we've been tracking our students for a number of years, and we've noticed that more of our students, when they graduate, they go on to do PhDs in biochemistry and biochemistry-related subjects. So this is continuously rising. A couple of years ago it was only 50%. Now about up to 60% go on to do full 3-4-year PhDs. Many of these will go on to work in industry. The pharmaceutical industries, the agri-food industries, they're packed with biochemists, immunologists, people who receive the type of training that we deliver. A number of them will go on to become teachers, and many will remain in academia and get lecturing positions, although they are far and few between, very difficult to get. About 10% will go on to do MSc courses. So these are graduates who aren't too sure what they want to do when they finish college. They might do a master's course in something slightly different to what they were trained in as an undergraduate. So there are the world is full of MSc courses, and lots of them are brilliant. They really are a fantastic way for students to figure out what they want to do just after they finish college. They can be really helpful. A number of our students, about 15%, go on and do medicine and pharmacy and these types of professional degrees. We've even had a couple go on to do dentistry. And then a number of students, about 10%, they just use it. Biochemistry it's a fundamental discipline. It's an excellent degree for training in logistics, how to tackle problems, how to get stuck into problems and produce outputs. And indeed, a lot of our students like the fact that it's a quantitative subject, and they go on. Once they finish college, they leave it behind, but they have been trained in a certain way, and they often go on and work in business, for example. We've got about 5%, the ones I say that we keep quiet about. Some of our graduates have actually gone on to be accountants. Now, I'm not sure why that happens. I don't like it. My colleagues don't like it. We're trying everything possible to screen these out before they come in and do biochemistry. I think we should actually get our psychologists involved and do some sort of Scientologist-type screening of our students before they come in. But they do, because again, it goes back to this fundamental training and quantitative. It's a quantitative subject. And it's very beneficial for people who want to, use that training and go on into accountancy. We've even had a politician. Now, here are some of our graduates listed that have come to mind. Liz Bonin, whoops, let me go back there. Liz Bonin, she's on telly all the time. She's a biochemistry graduate. She's on the BBC lots. Luke O'Neill is one of Ireland's most successful scientists. He's in our school. Keelan Shanley, again another journalist, RT primetime presenter, biochemistry graduate, and indeed often says her training in biochemistry made her what she is today. Now, I think she's just joking, but it just shows you it's an example of you don't have to become a biochemist after you do a degree in biochemistry. Sean McBrien went on to do medicine. He's a big GP practice down in Waterford. Albert McQuaid went into the food industry, Kerry Co-op, or Kerry Group. He runs the ingredients and flavours. Kerry is an enormous company, one of Ireland's, gold standard companies. Just a fantastic company, packed with biochemists and biotechnologists, because you have to be. The reason it is so successful is because they're cutting edge. They keep up to speed all the time with research that's being done. And a lot of their people are highly trained. Emer Maloney, she went on to do pharmacy. Brian Lockhart, he's one of the only non-French director of Servier pharmaceutical company in France, big, huge pharmaceutical company. Mickey Harrington, one of the leaders of the Waters Corporation. This is one of the world's leading technology companies in pharmaceutical industry and biomedical sciences industry. Jim Sullivan, head of drug discovery in Abbott, another massive pharmaceutical company, that American pharmaceutical company based in Chicago, but lots of places, a lot of people work in Abbott in Ireland. So if you do biochemistry and the related degrees, what you will receive is a training in fundamental biochemistry. And this is why it's so important for students, especially for students, they're only in as an undergraduate for 4 years. It's essential, if you want to go on and become scientists, that you receive the best fundamental training, and especially in this fundamental discipline that we call biochemistry. There's a big opportunity to study abroad. We're very keen. We send our students away to other laboratories in Europe, around Europe, also in the States, United States, on these Erasmus programs. Now, the Erasmus program is a European Union program which is fantastic for exchanging students around Europe. I was one myself back in 1990 when I was doing my PhD. My boss at the time, he said, you need to use a mass spectrometer. We don't have one here in the lab. There's one in Maynooth University, or there's one in the University of Siena in Tuscany in Italy. Before he had even finished his sentence, I was on the plane and in Siena for a whole year. Fantastic experience. And I would recommend, even if you don't do biochemistry or anything, once you get into college, look for these programs because they are brilliant. You learn, well, I was about to say you learn languages. I still can't speak Italian. I can eat pasta and drink Chianti, but my Italian isn't great. Other universities and industries as well. So we send our graduates to industry. They go out for 3-4 month summer projects, summer student projects. And this is really, really essential. This is where it's vital for students to get experience in industry outside universities. Because after all, that's where most of our students will end up. So we've got a number of pharmaceutical partners, industry partners that our students go in and out of and learn, start off learning a bit of the trade, what's involved in a pharmaceutical company or so on, or in a biotechnology company. We're trying to increase this as much as possible because this really will help our graduates get jobs straight away. And of course, we have top research labs here in Trinity, in the School of Biochemistry and Immunology. And some of Ireland's leading labs are here. It's the biggest biochemistry place for biochemistry in Ireland. A lot of our students get snapped up places like Cambridge and Oxford, Harvard and so on to do PhDs. Mind you, a lot of them stay here because the opportunities are so great. You can read more about, or you can hear more about the type of research that we do. We run a public Biomedical Frontiers lecture series. And you can, if you go to the TCD website, the biochemistry website, these are held every 3 weeks. We just started them this year, and they've been massively successful. There's about 300 people come every 3 weeks. And the fourth one is happening in a few weeks' time. So these are the courses. Biochemistry degree, so this is part of the TRO 71 stream. So you come in, you do general science for a couple of years. You have to do biology courses, that we insist if you want to do biochemistry. So when you get into third year, you choose whether you want to do biochemistry or immunology or molecular medicine. So we have about 60 students that will graduate in these degrees. And these students will be placed in labs. They'll do final year research projects with some of Ireland's leading scientists. And we also are involved in this one here, TRO 56, the Human Health and Disease that we run with the School of Medicine. But a lot of biochemistry in first year and second year and in third year that these students take. Why is biochemistry so important? It's, as I said, it's the fundamental discipline in medicine. Not just in medicine, but if you want to understand organisms, you have to understand the biochemistry. Most of the Nobel Prizes in, say, physiology and medicine, indeed a lot in chemistry, they've been awarded to biochemists. Biochemists, they discover molecules that control life, basically. It's as simple as that. It is the most fundamental discipline if you want to understand how organisms live and die. And this is why, if you go to any pharmaceutical company in the world, it's packed with biochemists. You go to any hospital, any research facility in hospitals, packed with biochemists. You go to departments, departments of genetics, microbiology, neurosciences, packed with biochemists. And it's because the biochemists work at the fundamental basis of life. We discover molecules that communicate with each other. We figure out how the structure of these molecules, why the structure is so important, that allows drugs to be developed to interact with these molecules. It really is as simple as that. Well, it's not actually that simple, but that's where we're working at. That's what we do. And it's not going to change. It's an old term, biochemistry. It's very simple. It'll be around for hundreds, thousands of years to come. It is really, this is where it's at in terms of discovery of molecules and how they control cells and give rise to disease. So let's go into a bit about the types of research that go on in my school. We have a lot of immunologists. So this picture here, these are two immune cells interacting with each other. It's a beautiful, beautiful photograph. And immune cells, as you know, have this fantastic ability to recognize foreign invaders and to go in and kill them and to activate other immune cells and to send out antibodies that fly around the body and prevent us from getting sick. This, I'm sure you've all seen in various textbooks, school books, biology books. This is what I was introduced to as a student. This is a cell, nice spherical shape. It's got a nucleus inside with the DNA. It's got mitochondria, the powerhouses of the cell that make ATP. It's got proteins. If it's a plant, it's got a chloroplast. It's got the Golgi, which secrete proteins and so on. And that, to me, was fascinating. Of course, there's nothing like that. It's much more dynamic than that. All these things are flying around. There are thousands, there may be 30,000 genes. So this is where it was fantastic that the human genome has been sequenced. There are only about I think it's 35,000 genes that give rise to over 100,000 proteins. So you can get maybe 130,000 proteins, and they can be clipped up. And all these proteins can have multiple activities. And then these proteins can come in and touch and communicate with each other. And you get more, you get a different activity. This is really where it's at. It's trying to understand how these proteins communicate with each other. And to control that, because that's ultimately what controls disease. Not just disease, it's normal cellular function. It's vital that we understand that. These cells exist in the brain. They're not shaped like that. They're much more fantastically shaped, like neurons and so on. Same things inside them. all these biochemical molecules flying around the place. We need to understand how they communicate with each other and control normal function, and of course, abnormal function. Now, to enable us to understand proteins, now genes code for proteins, it's these proteins that are vital. We need to understand the structure of these proteins. And this is where it's really, really difficult. But we're quite lucky here because we've invested a lot of resources. Trinity has invested a lot of resources in recruiting scientists that are expert in solving protein structures. So here, what we do is we purify a protein, and in order to get the structure, you fire in X-rays, but you have to have it as a crystal structure first. You can't, if it's a protein in solution, you won't be able to get any meaningful information in terms of this type of structure. You can get information for sure, but not the actual crystal structure. And then you can fire it, bring it over to the synchrotron in Grenoble, this massive facility that speeds up electrons and X-rays and fires them into the proteins, and the structure can be solved. And then once you know the structure, you can say, well, okay, here, right here in the middle of this protein, that's the active site. Let's design some drugs that will get in there and activate the protein or inhibit the protein. And this is how drugs are developed. Vitally important to understand all these protein structures. And indeed, that's what the majority of biochemists are doing around the world at the moment, trying to figure out the shapes these proteins take when they're inside the cell, when they're outside the cell. And the shapes, sometimes they're misfolded, like in Alzheimer's disease. There's lots of misfolded proteins in the brain that are part of the whole degeneration of the brain. Vital that we understand these protein structures. I'm from the School of Biochemistry and Immunology. So we're all biochemists there. A number of us specialize in immunology, in the immunology discipline. So you'll find that there are some of the world's leading immunologists based here in Trinity who are making fantastic discoveries. Some work in the area of influenza virus. This is just an amazing photo. These are amazing photographs, really. This is the influenza virus. And as you know, the influenza virus can get into the lungs and attack the host cells. And there are enzymes, proteins, biochemical molecules that splice in the influenza viral DNA into the host DNA, and the host DNA or the host cells make more influenza, and then that comes out as an aerosol, and it's passed on from person to person. So we need to understand, to understand how this happens, you need to understand enzyme structures, protein structures, these molecules of life and death. For example, this one here, neuraminidase, is an essential protein enzyme that the influenza virus has here. It allows the influenza virus to penetrate into host cells. Neuraminidase, it breaks down sugar molecules that are present on the host cells and allows the DNA, the foreign DNA, to get in there and to be replicated and a new influenza virus made. Also, hemagglutinin, this one here, is very important for cell binding, also present on the outside of the influenza virus. Biochemists would have developed Tamiflu, for example, is an inhibitor of this enzyme here. Vaccines as well. Now, the whole world of vaccination is, as you know, vitally important to human health. It's one of the reasons, the basic reasons why we're living so long now, because we're able to vaccinate our children. Massively important. One of the most singly important discoveries, vaccination. It's obvious, really. So this is a target. Vaccinate against this protein here, so that once it's made, the body's immune system immediately recognizes it, that it has antibodies. It gets in there, binds to it, and then the body's immune systems come in and destroy the influenza virus. Works very well, except the influenza virus is just, these are antibodies. This is the structure of an antibody. And these regions here recognize, for example, the influenza virus. But the influenza virus is it's a really special organism because it has this ability to mutate all the time. The cell surface mutates. You've got all these different sugar molecules on the cell surface attached to proteins. It's constantly evolving rapidly. And this is to do with genetics. The mutations are introduced into the DNA that codes for the neuraminidase protein, for example. So it's very, very difficult. And as you know, the influenza virus often doesn't work. The vaccination doesn't work. It might prevent a certain form of influenza, but it's rapidly evolved or evolves, and then the next bout or epidemic of influenza, our systems can't deal with it. So it's vitally important to be able to understand the biomolecules that allow this, what we call antigenic drift, to materialize, and then to develop vaccines rapidly that can remove them. So all the big pharmaceutical companies work in these areas, nearly all of them. And so do a lot of our scientists. Now, I'm sure you're well aware of what's been happening. In such a short period of time, we're experiencing rapid evolution of these organisms. And we've got things like avian virus, swine flu virus, and human virus that I've already spoken about. These things in little pockets around the world pop up every now and again. And they're interconnected. They can certainly, in models, in labs, you can infect one organism with another organism. And so these things are constantly evolving. And as they are introduced into different species, they change another little bit. And there's this rapid evolution of these viruses. Very, very dangerous. And indeed, the swine flu, what is emerging are these mixtures as well of viruses. Now, what hasn't happened yet is where we get widespread transmission to humans. So obviously the people like the World Health Organization keep on this as much as they can, on top of it as much as they can. Because if there's something, I wouldn't be so scared about, atomic warfare as I would with these types of viruses. These are they have the real potential to cause massive destruction to the human race. So it's vital that biochemists can get in there and understand how these systems work and develop new vaccines, predict how the organisms or the species are going to evolve, and stop it before it actually happens. What you shouldn't do is this. Not one of my offspring, but anyway. Now, hepatitis C virus. We have a couple of groups in our school that specialize in this type of research. Here's a healthy liver on the left. Lots of hemoglobin in there, lots of red, iron. Here's a hepatic hepatitis C liver. So you can see big breakdown, a lot of fatty acid deposition, tissue breaks down. The hep C virus is notoriously very, very dangerous. And if it gets in and it's uncontrolled, this is what happens, and the person will die eventually, or a liver transplant is required. We're talking about molecules of life or death. Some of our researchers, Cliona Farrelly and Nigel Stevenson, in fact, Cliona will be giving the next Biomedical Frontiers lecture in a couple of weeks' time. Look it up on the web, as I said, on this area. But what they discovered was that this STAT3 protein, it's a molecule of life and death, really, life for mammalian cells and death for viruses. So what they discovered in healthy liver cells, here you can see in the green, we've got this STAT molecule. We can actually observe these single molecules under a microscope. We can attach these little fluorescent tags to them. It's quite amazing, really. And in livers that have been hit with the hepatitis C virus, this green molecule is lost. The signal for this molecule is lost. They can bring it back by inhibiting this thing called a proteasome. And it brings back the STAT3 protein, and the cells remain living. They don't die. So what they've discovered is this interaction with the proteasome that controls whether the hep C kills the liver cells or not. And this is really this is the cutting edge of biochemistry and structural biology. This is generated from another guy in the school, Amir Khan, who runs the X-ray crystallography group. So he was able to generate the structure of proteasome. All these little ribbons here, these little helical structures, these are proteins. Lots of amino acids joined together that form these fantastic shapes. This barrel, the proteasome is where all our proteins are broken down. Proteins that are slightly damaged or no longer needed. The cells turn them over, and they put them into these proteasomes and break them down. Now, what Amir did here was he was able to model the proteasome, and this is the STAT molecule, the STAT protein, and he's able to show where it goes and it binds in here, moves in here, and interacts with the proteasome in this manner. So it's this type of information that's vital to come up with new drugs that stop this process from occurring. And this is really what drug companies want. What we as biomedical scientists want is to make this type of discovery. Rachel McLaughlin, she works on Staphylococci aureus bugs and others as well. MRSA is this methicillin-resistant one, which is, as you know, very dangerous in hospitals, especially in Irish hospitals as well, because although we have been improving cross-contamination and cleaning hospitals properly and getting doctors to wash their hands, simple things like that, will have decreased the incidence of MRSA, but it's a real killer of a bug, especially when it gets into young kids, premature born kids, or the elderly population, very susceptible to MRSA. We've all, I can't remember that something like 60 or 70% of us have MRSA in our nasal cavities, but it doesn't really affect us. But there are certain sections of the population where it really does. So it's very, very important to understand how these bugs avoid our immune system. And this is where all our immunologists come in. They work at this level. They say, okay, what's wrong with that person's immune system? Why does that person's immune system not kill the MRSA bug when it comes in contact with them? So these are the big questions. And to do that, you have to understand, go inside the body's immune systems and look at all the hundreds of thousands or the thousands of different proteins and see which ones are different, how some communicate differently, what's special about them. A lot of work to be done. But we'll get there. So it's these bacteria here. You can see them. They're tiny. This is the tip of a pin. But our immune system is fantastic at recognizing them and activating themselves. So our immune cells become activated, and they send out antibodies that bind to these bacteria. And then we've got natural killer cells that come along and engulf the bacteria and get rid of them, or macrophages that come along and engulf them and chew them up and get rid of them. It works brilliantly most of the time. When it doesn't work properly, that's when we get a lot of diseases. So here you can see this is an immune cell, and it will recognize, it sends out tentacles. Now, this looks fantastic. It's a fantastic picture. You can see it on the outside of the cell. But in order to do that, the immune cell has to recognize the proteins on the outside of the bugs. And then it recognizes certain sequences of proteins, certain protein structures on the bugs. And then it sends out these tentacles and pulls them in and engulfs them and chews them up and spits them out. So there's an amazing repertoire of immune cells around the body that take care of us, except rheumatoid arthritis, multiple sclerosis, Alzheimer's disease, lupus, chronic irritable bowel syndrome. All these things are diseases of the immune system. And it's vital that we figure out the molecules that control whether something lives or dies in these diseases. So lots of work going on in this area. Cancer biology. Of course, we're going to have researchers working on cancer. It's so prevalent in society, especially as we live longer. Cancer is an age-related disease. It's one of the biggest risk factors for cancer. So the longer you live, the more likely you are to get cancer. Well, the longer you live, the more likely you are to get a neurodegenerative disease as well. But we can keep people living longer But associated with that is this risk, big increased risk for getting cancer. So cancer cells rapidly divide. Now, there are certain molecules inside cancer cells, biomolecules, that allow cancer cells to do this. And the cancer cells, they're amazing. There's amazing biology going on inside them. And it's at this interface where a lot of our researchers are working. They're trying to figure out what makes these cancer cells so special. Why do they thrive on, say, glucose, for example? Why do the cancer cells are brilliant at getting glucose, sucrose, and extracting energy from them and replicating, really rapidly. These are the kind of questions that our researchers are trying to answer. Also, cancer cells avoid the immune system. A lot of the time, we don't know it, but we get cells replicating in our liver or around our body, and some of them replicate badly. There's a mutation introduced into them. But we don't even know it, but our immune system clears it up. Our immune system can recognize cancer cells and tag them and label them for destruction before they get to a tumor size. Except it doesn't always work like that. Obviously, the immune system doesn't do it efficiently all of the time. That's an area, where a lot of our researchers are working at, trying to figure out how we can improve the immune system to get rid of cancer cells the moment they're formed. One of my colleagues, Ken Mock, works on this Hamlet protein, which is a fantastic name. Human alpha-lactalbumin made lethal to tumor cells. Hamlet. Now, this is an amazing discovery, really, from breast milk, believe it or not. This is a protein, alpha-lactalbumin. This is the kind of shape it has. You can extract from breast milk. And in the presence of this lipid or this fatty acid called oleic acid, it undergoes a change in shape, different shape to it. But they discovered this amazing activity that it kills cancer cells, but not host cells. And the types of cancer cells that it kills are these glioblastomas. So glioblastomas are really nasty tumors that you find in the brain. The brain is made up of different types of cells, a lot of neurons, but also a lot of support cells, immune cells. Sometimes when these replicate, it's not controlled and they develop into glioblastomas. Hamlet protein is really, really good at killing these cells. So you can see here's the cell, it's given Hamlet, and it starts to die by a process called apoptosis, which is called programmed cell death. All our cells have this ability to die in a controllable manner. Our host cells, our liver does it. I'm happy that my liver is constantly, millions of new cells are dying every day, and millions of new cells are being made every day. It's a very important process to control that. It's this process of apoptosis that allows cells to die in this controllable manner. Of course, in something like stroke and a brain attack, it's the opposite. You get necrosis. These cells burst open. There's massive damage. But it's the apoptosis that this Hamlet protein can initiate in these cancer cells, but it doesn't do it in the normal cells, in the host cells. So a fantastic opportunity here to make major discovery, or in the clinical setting. Now, this is from my group. A lot of the researchers work on neurodegeneration, Parkinson's disease, the second most common neurodegenerative disorder. You see it's the shaking. It used to be called the shaking palsy. Somebody might have that kind of shaking, or very difficult to walk, stupid posture. Generally happens over the age of 55, 60. There is a young onset form. Michael J. Fox, the actor, would have had it, or has it. But the majority is over the age of 60. Age-related disorder generally. And what happens is you lose these neurons here that go, these are the dopamine-producing neurons. Dopamine is a very important neurotransmitter in the brain. There are 10 to the power of 11 neurons in a typical adult brain. A few politicians might forget about them. They're about take off a factor of 10. But in the normal population, 10 to the power of 11. Now, each of these neurons can connect with other neurons. You can get up to 10,000 connections. One neuron can have up to 10,000 other connections with other neurons. And then those connections can change in strength. It's like a handshake, a really strong handshake or a weak handshake. So there's this plasticity involved. in Parkinson's disease, we've only got about 350,000 dopamine neurons. Not that many, but they're essential because they start here just above the brainstem, above the spinal cord. When the spinal cord comes into the head, just above that is the brainstem region. And these dopamine neurons die off. It's these ones here that are lost. And they're right at the start of this network. And the end result is, the brain there's a balance of inhibition and excitation. But if these neurons are lost, the end result is that we don't have control over our muscles. The classical symptoms of Parkinson's disease. So what we've been doing in the lab is using stem cells to develop, to change into dopamine neurons. And we've done this a number of ways. This way is, a few years ago, some colleagues in Japan discovered this most amazing revolutionary technique where they discovered they were able to add some biomolecules, 4 little biomolecules, biochemicals, to skin cells, adult skin cells. And these skin cells became reprogrammed cells. Now this is, when I was in the 80s and 90s being taught biochemistry, everything goes forward. It goes from a stem cell forward into a neuron or into cardiac cells. Nothing ever goes backwards. That's what I was brought up on. These guys in Japan recently got the Nobel Prize a few weeks ago for it, and discovered that by adding these biomolecules, these 4 biomolecules, they were able to instruct the skin cell, which was it's a differentiated cell, to go backwards. It becomes something very similar to an embryonic stem cell. And as you know, embryonic stem cells have the potential to change into any type of cell tissue in the body. And this is what we're able to do now. We're able to take skin cells, introduce these biochemicals into them. They reprogram, they become something very similar to a stem cell, and then we can add factors, more biochemicals, to encourage them to become dopamine neurons, dopamine-producing neurons. This is where the technology is at. It's now possible to do this. Now, we're not at the stage where we're able to transplant these neurons into the human brain yet. Some people have tried, but not. There's been a bit of success, but we're getting there. So amazing, amazing discoveries to be made in this area, and you'll hear more and more about this type of research as the years go on. Now, just to finish up, there are lots of other types of research going on in the school, but roughly we've got over 200 researchers, 26 group leaders. So these are principal investigators, we call them. They lead laboratories. About 80 postdocs, so these are people after they've trained as PhDs, after they've got their PhDs, they become professional scientists. About 86 PhD students, 60 final year undergrads, but we actually teach about just under 20% of all college students. That's medical students, pharmacy students, nurses. They all come through the biochemistry teaching lab. We teach them fundamental, practical skills. So it's an enormous contribution to teaching Trinity students in general. It's that fundamental. These students have to be taught the basic biochemistry techniques, and that's what we specialize in. Lots of research funding coming in, etc. These are the types of equipment. This is amazing equipment for separating these immune cells. Here we've got nuclear magnetic resonance for solving protein structure. Here we've got tissue culture for doing the stem cell type stuff. These really powerful microscopes for looking at cells, looking inside cells. And this is where it all happens now. This is Trinity's flagship Biomedical Sciences Institute. It's just beside Pearse St train station. We moved in last year. This is it here. And it's a huge, huge building. The School of Biochemistry and Immunology, that's where the majority, we're based there, we're the anchor tenant. All our labs are there now. We used to be all over Trinity, but now we're together as one. But what we've done is we've incorporated the bioengineers. They're a part of the institute. Because it's really important to develop new medical devices to deliver stem cells into bodies. You can't just inject stem cells in. You need to have biomaterials that allow them, that facilitate them. So the bioengineers are there. We've got clinicians. It's very important to translate our discoveries into the patient. So we've got clinical scientists based there. We've got pharmacists to develop new drugs. So we solve a structure, we come up with a new target for cancer. We go to our chemistry colleagues, organic chemistry colleagues, and our medicinal chemistry colleagues, and say, can you make a drug? Here's the structure. Will you make a drug? And they say, yeah, we'd love to. We've got something to work at. And that's the way it happens. And then who else have we got? We've got, that's pharmacy as well. I'll mention them. So all of these have come together to the Trinity Biomedical Sciences Institute, where we're based now, working on major diseases, human diseases. And again, just there, you can see get the list of the lectures that are open to the public. There's some great, great lectures. So I'd encourage you all to come in. They're generally held on a Wednesday or Thursday evening. And with that, I'd be very happy to answer any questions. Thank you.